So I just wanted to post this lecture as a quick review, just to clarify one or two things about primarily the domains that we're going to go over in this class. So think about the following questions for a moment. What is a prokaryotic cell versus a eukaryotic cell? How can you differentiate the two, compare and contrast? Think about what are the three domains, how do bacteria and archaea differ? So let's kind of delve a little more into that to build on our first sort of lecture for the course. So at a glance, we see some differences between eukaryotic cells and prokaryotic cells. Now, all cells have a plasma membrane, some kind of barrier to the environment. Also, they contain some kind of cytoplasm, some kind of solution of molecules and salt inside the cell. Also, some form of DNA genome, as well as some kind of protein synthesis. So that's just kind of talking a little bit about all cells, but we also can see some differences right off the bat between looking at the levels of complexity between eukaryotic cells on the right there and also on the left there, prokaryotic cells. So there are some similarities, but there's also some stark contrasts or differences. What are those differences? Well, we have prokaryotic cell here on the top, does not contain a nucleus and does not contain membrane enclosed organelles. And for eukaryotic cell on the bottom here, membrane enclosed organelles, largest is usually the nucleus. You also see a lot more complexities or differences in size. So just right off the bat, we have some ways in which they're similar, but really more some ways in which they're different. So think to yourself, what are those three domains of life? By now, we should all know this. This will be asked on every single examination. As is the study of biology. What is the study of biology? The study of life. What are those three domains of life? So in your mind, you should be thinking that the answer essentially is the domain eukarya, domain archaea, and domain bacteria. Now, domain bacteria, domain archaea, domain eukarya. We see here on the top, bacteria and archaea have prokaryotic cells or are characterized by having prokaryotic cells. And then obviously on the bottom there, the domain eukarya, the group that we're going to spend more time on this semester, characterized by eukaryotic cells. Those are the kingdom Plantae, kingdom Fungi, kingdom Protista, and kingdom Animalia. So for purposes of exam 1, know those 4 kingdoms underneath eukarya, and we're going to start getting to know those kingdoms quite well as we proceed forward in the course. So once again, the domain bacteria, just to cover that a little bit more, the first major group of prokaryotes, characterized by having strong cell walls. Very important for a number of reasons. That includes soil enrichment or nitrogen fixation. Also, E. coli in the human gut helps synthesize vitamin K. Oftentimes you see bacteria colonizing dead organisms. Also for medicine in terms of producing antibiotics and insulin, so very important from that aspect alone, humans at least, and does contain most modern-day prokaryotes. A couple of things to note in addition here, just to kind of reemphasize this idea from this table in your text. The domain bacteria also characterized by having cell walls with peptoglycans. Also the membrane having a lipid bilayer. They are sensitive to streptomycin and they are able to undergo the Calvin cycle. So kind of just think about that as we compare the next group. Now here are some examples of bacteria. So you're going to get into this more if you ever take microbiology. There's a great number of bacteria that we're not going to have time to cover in this course. A lot of really interesting things are happening with human health and the microbiome. So we might have an article too about those later in the semester. But just to give you some idea of different diversities within bacteria as a domain. Once again, there is a great deal of diversity in different groups, different phylums within bacteria, but we're not going to focus on that in this course. We just won't have time. So moving forward, thinking about the domain archaea, we tend to find these in very extreme environments, so that's oftentimes why they're called the unique condition prokaryotes. They also do have unique cell walls. We tend to think about these as being either methanogens that produce methane, extreme halophiles are living in high salt concentration or areas or environments or habitats. Also extreme thermophiles, so high temperatures, so interesting areas in which they're inhabited. And also, they're thought to have split from bacteria about 2 billion years ago. And now, something else to note in terms of the domain archaea, in terms of their cell wall, they have no peptoglycans. Their membrane, the lipid bilayer or monolayer, so there's some diversity there, where they are resistant to streptomycin and also no Calvin cycles. Just a couple of ways in which we can compare and contrast bacteria and archaea in terms of domains, what they have and what they don't have, where they're found in terms of their diversity. This gives you some more indication in terms of what I was mentioning in terms of some of these being methanogens, what they're found in different habitats. So there is some different phyla that are within archaea. So how do these two domains differ, archaea and bacteria? So here's a table from your text, and so I just want to reemphasize this idea because we didn't really hit upon this in the first lecture, so I want to revisit this before exam one. In terms of how they're different, how they're similar, they're obviously both prokaryotic. They have a variable cell morphology. So take a look at this table and think about maybe make a list of 2 or 3 items in terms of how they're different and how they're similar. So I just want you thinking about not only prokaryotic and eukaryotic cells, but also if you were comparing contrast, perhaps bacteria and archaea, what are some similarities? How are they different, more importantly? So compare and contrast the domains bacteria and archaea because they are unique, distinct domains. And we are going to really just focus on them a little bit in this first bit of the semester for exam one, that we're going to move forward and really focus more on our next group. Okay, so like we mentioned, we're going to focus more on the domain eukarya in this course. And so we've already talked a little bit about fossils. And there are 4 kingdoms within the domain eukarya once again, so know what those 4 kingdoms are. Animalia are animals, fungi, plantae, plants, and then protista, protists. And so we'll go over those as we proceed in lecture. Of course, these are characterized by having eukaryotic cells, true cells, with some level of complexity, larger size cells, and also a lot of membrane-bound organelles. So a couple of ways in which we're going to focus more on this domain, a couple of ways thinking about eukaryotic versus prokaryotic cells in these 4 kingdoms of the domain eukarya. Okay, so we do have an exam coming up on Canvas, which will require the same respondents, lockdown browser, webcam as previous quizzes, so be aware of that. Make sure you go through and review the various different modules. Make sure you participate in this week's discussion for review. And one thing would be really good to have a good understanding of the content would be to go through those learning outcomes, maybe quiz yourself on those learning outcomes, answer them off the top of your head. Take a blank piece of paper, start answering those, or do the same with this week's discussion. The videos are all there for review if you need to look at lecture notes. The exam will largely come from the lecture material, but we'll also include some questions from the various videos or articles, which all sort of add to this body of knowledge for exam one. So there are a lot of options for how to prepare for exam one. So go ahead and do that this week and the rest of next week, and good luck and have a great week.